everyone, welcome back to the cell signaling course. We are going to learn very new type of receptor mediated signaling in today's class, and that will be G protein coupled receptor mediated signaling. In the last class or until the last class, what we saw was different types of receptor which pass the membrane only once or maybe twice. But here these receptors are very unique in the way they pass the plasma membrane. We will see how these signaling events mediated by GPCRs are very different from what we have been seeing for the other set of molecules. You may see that there are multiple final effects which are happening downstream of GPCR, look similar to other signaling, but the intermediates which are involved, the effectors which are involved, they are going to be rather different in the GPCR signaling. So let's start with the GPCR. As the name suggests, GPCR stands for G protein coupled receptor. When in earlier classes we were discussing about the receptor types, we briefly introduced what exactly GPCR receptors are and how do they signal. But let's see what we have what we understand from the GPCR. These are also called 7-pass transmembrane receptor because the receptor, if this is your plasma membrane, it crosses the membrane 7 times. So they are basically alpha helices. Each of these pass to the membrane is by 1 alpha helix. So there will be total 7 helices which will be crossing the membrane. Since it has 7 helices, this is also called heptahelical receptor. And also going by just simple appearance, that how many times it is going around the plasma membrane and coiling, is also called serpentine receptor. More importantly, this represents a class of receptor which do not have a direct enzymatic activity. There might be some hidden enzymatic activity, but there is no direct enzymatic activity. Thus, they are dependent on enzymes which communicate with these receptors once the receptor is activated. And then there are these heterotrimeric G protein, because of which they are called G protein coupled receptors. They associate with the receptor once the receptor has bound to ligand, and these activated G proteins now take the signaling forward. So let's understand more about these GPCRs. So among different types of receptors which we are discussing, in human, this is most abundant type of transmembrane receptor. It has, as we say, all the molecule which are falling in the category of GPCR, they pass the membrane 7 times. The structure is more or less fixed that they must pass the membrane 7 times. You can assume the number almost 4 to 5% of the human genome codes for GPCR. Forget about the other molecules which are involved in the GPCR signaling. Just the receptor itself is represented 4 to 5% of the human genome. Then these GPCRs are classified further into 6 different classes based on what exactly is their ligand, which process they participate in, and what eventual signaling they mediate. Many of the small molecules, hormones and amines, biological amines, which is like some neurotransmitters and so on, they are the signal for these GPCRs. When I say small molecule, there could be any metabolite which is generated into the system or something which is present in environment or made by some other living being that is being serving as a ligand for these GPCRs. Also, it is very important to remember that many of the sensory organs which are responding to availability of cues is via GPCR. We have seen until now that how receptor serine threonine kinase or receptor tyrosine kinase or Notch delta signaling, they are important for survival, growth, proliferation, development and so on. Here what you notice is that basically GPCRs are largely involved in responding to the environment and regular normal growth. Basically, like more to the sense organs which we through which we are perceiving different sensory stimulus. Then these receptors, among the multiple GPCRs which are known, there are good number of them which are associated with olfaction. The olfaction is the process of smelling things. So multiple different molecules could be there which will be serving as a ligand to one particular GPCR. Still, there are so many GPCRs, since the number is very large, there are so many GPCRs which have still not been characterized in the sense that what is the actual ligand for those GPCR. Such receptors are called orphan receptors. So you realize that there are so many differences which are there in case of GPCR when you compare to any other receptor which you have studied until now. So we can say that GPCR signaling is mediated by these serpentine receptor, and these receptors associate with enzymes. They do not have their own enzymatic activity. So basically, I have mentioned this briefly in one of the earlier classes, that if you talk about receptor and you talk about an effector molecule, think about an effector molecule like this. In this case, consider G protein. Here the G protein which is involved is a heterotrimeric in nature, so it will have alpha, beta, gamma subunit. So the association of the receptor and the G protein, it's not like that it does not happen in absence of ligand. If this ligand is not available, it does not mean that this association is not happening. It is there, just that it is not strong enough, because the association of this, generally G alpha is present in GDP bound state, and in GDP bound state it is not at all associated, or it is associated with the receptor and it is not signaling. So when ligand is not present, you do have this G alpha associating with the receptor, but the transience time and the strength of interaction is very poor, because there is a preferred route of receptor interacting with G alpha protein in GTP bound state. So dynamic association of G protein with its receptor is basically continuing the GPCR signaling. Also, we have seen in multiple other signaling that once this kind of effector is bound to the receptor, signaling continues, and we have to stop the signaling at some point of time so that we can bring the situation back to normal, mimicking a condition where ligand is absent. Because we also realize that ligands, they are available, there is a peak of ligand which will be available, and then slowly it will taper off and the concentration will go down, below which there will be no signaling going on. Now if we talk about any signaling, and let's talk in case of GPCR. So there are multiple requirements of a signaling process. We have seen that you need the ligand, you need receptor, and you need the signaling molecule, effector type or adapter type. In case of GPCR, the receptor has very unique arrangement. We have been seeing this, that so this is 7 pass transmembrane. If you think about this is the orientation of helices, then basically it will be connected this way. So, and then intracellular C-terminal region and extracellular region which is N-terminal. So, there will be different features which will be associated with that. Now, what you can clearly notice that there is extracellular region. Here there are 3 loops, loop 1, loop 2, loop 3, and internally there are 3 loops which are intracellular loops. These are extracellular loops and these are intracellular loop. what is very obvious, that ligand binding will happen in the extracellular loop region, and effector molecules will be binding somewhere here. One of the first effector molecule which comes downstream of these receptors is G alpha protein. Okay. So, this G alpha basically will occupy space in the intracellular loop region. So, basically when signaling is happening, somewhere this helices has to rearrange to make space for this G alpha to come and associate. Extracellular loop region, you can understand that loops are going to be more or less similar. So, what induces the specificity towards one ligand? We saw in other cases of other receptors that there are multiple different types of extracellular domain which was allowing the binding of ligand. Here it seems that there are some sort of loops which are present which is giving the specificity towards the ligand. Further, there will be these helices are embedded in the membrane, so they are arranged in a particular fashion which allows the signal to be buried in this extracellular loop region and binding to that. So, in essence you have the, if I say the helices, so such 7 helices will be arranged to give space for some molecules to bind, which could be eventually towards the C terminus, you will be seeing the signaling happening. There were a lot of signaling which went on in previous cases where C terminal domain was allowing a docking site serving as an adapter for next molecule to come, bind and activate signaling. We will see here that how C terminal region is going to play a very important role when it comes to terminating the signal. So, if we talk about other simplistic features of the GPCR, we can assume, if I can say that these are cylindrical alpha helices. So, they will be having a specific orientation and arrangement when receptor is unbound. There is no ligand which is bound. So, this extracellular region, we have already said, we are talking about this as the membrane in which this is embedded, and you will have the ligand here, and here you will have the regions where G alpha will come and bind. We will come to G alpha because we know that this is another critical requirement for GPCR to function. We are discussing now the features of these GPCRs, and it seems that there are only 2 main features over here: the loop regions which are allowing the ligand binding and the G alpha binding, and then these free domains in the C terminal region which is for the receptor deactivation. Because these membrane spanning helix regions are more or less going to have very unique features which are important for spanning the membrane. Now, if we look at the more into the structural features in detail, I mentioned it to some extent, but remember they need to be regulated tightly. So, if I can just say helixes, so each helix is spanning the membrane. Generally its N terminal is outside and C terminal is inside. Here in between the helices you will find the connecting loops, extracellular loop 1, 2 and 3. They will be arranged such that the ligand can be accommodated. Intracellular loop will be arranged such that G alpha can be accommodated. And the C-terminal tail, also remember when we are talking about this entire thing, G alpha I am talking about, but we are talking about heterotrimeric G proteins. So, entire G alpha, beta, gamma come together in the process of receptor activation, and once the activity starts, once this G alpha moves from GDP bound state to G alpha GTP bound state, it no more binds the beta gamma subunit. If you recollect, in one of the previous classes I mentioned that how small event actually increase or decrease affinity for two proteins coming together, like here G alpha and beta gamma subunit, they are together as long as G alpha is in GDP bound state. But once the receptor is activated, G alpha has come into vicinity, you will see G alpha going into GTP bound state, and that is where it will lose affinity for beta gamma subunit, and that will be moved aside, right. One very important thing which you notice here is that G alpha is coming as a GDP bound molecule, but it is going into the GTP bound state. And if you recollect the G protein things which we discussed, any G protein which is in GDP bound state, for going it to GTP bound state, you have to first dislodge the GDP and place the GTP. And this process is mediated by proteins which have GEF activity, guanine nucleotide exchange factor activity. So, it seems that in these GPCRs, the GEF activity is somewhere in this intracellular loop region, so that when GDP bound G alpha goes inside, the exchange happens and G alpha converts to GTP bound state and it is active. Also remember, the GTP bound state was considered on state of the G protein. So, here also this becomes on state, and this is now going to amplify the signal downstream of the receptor. Now, if I just show you how simple changes happen which are required for activation to happen. So, basically what I told you in the previous slide, that G alpha goes into GDP bound state and comes out as a GTP bound state. So, if we take the cross section, if I just simply make a diagram here saying that this is your 7-pass transmembrane, and I am asking you to, I am trying to present a view which you will get by cutting it and looking from top. So, basically top view. So, now think about this scenario where we have 7 helices. So, this is a cross section which we are seeing from top of the top view of this cross section of these helices. So, if I say helix 1, 2, 3, 4, 5, 6, 7. So, the helices, for example's sake, if you consider that the intracellular loop region of some of these helices are engaged in G alpha binding. So, in this example, for example's sake, in this case we are seeing that the alpha 3, alpha helix here, alpha 5, alpha 6 and alpha 7, they are having the region which should bind to G alpha. They will coordinate the G alpha in this area. When ligand is absent, they have an orientation where there is not enough space, because of which the G alpha cannot bind, right. Now, consider a situation where ligand has bound, which has actually made changes into the helices, their position, relative position. And what happens now that you have 1 helix, 2 helix, 3 helix, 4 helix, 5, 6, 7. There is a small rearrangement which has happened into the helix. So, you can notice from here to here, this is 1, 2, 3, 4, 5, 6, 7. Now, the region of this helix 3, helix 5, helix 6 and 7 is so nicely oriented that G alpha can be coordinated very well. So, Small change which is happening because of the ligand binding and rearrangement of these helices allows G alpha to be tethered. You have to remember that in this scenario, G alpha was not spending enough time in the vicinity of receptor because the space was not available. That is why it could not be activated. But when ligand binds, the rearrangement of these helices allows a space to G alpha to bind and thus get converted into G alpha GTP bound form. And this is how it will, once it is activated, remember G alpha, beta, gamma in GDP state, if I put it here, they are together. Once G alpha moves into GTP bound state, it separates from beta gamma subunits. And if you recollect in the previous one of the previous classes, when we are discussing type of receptor and activation, G alpha has its own membrane binding ability. Out of the beta gamma has the membrane binding ability. So now both the parts of these G alpha beta gamma heterotrimeric complex are tethered to the membrane. They both can participate in the signaling, or only the G alpha is participating in the signaling. Now let's see further the other component which is required. Until now we saw how the prime component of GPCR signaling, the receptor itself, what are the features of that and how it undergoes rearrangement and so on. The next very important molecule here is G alpha. So if you recollect early discussion, G alpha into GDP bound form goes to GTP bound form. This process is catalyzed by Jeff, and when this molecule has spent enough time activating the process or meeting the requirement of generation of this triphosphate bound form, it goes back to the diphosphate bound form, and simply what happens here is the hydrolysis of GTP happens, and for that you need a class of molecule which are called GAPs. So guanine nucleotide exchange factor is activating the system, and guanine gap proteins, they are GTPase activating proteins, they are actually making it in the off position. If you think about, I told you there are 800 genes which are coding for GPCR. So how many genes, how many different types of G proteins are there? So there are broadly 4 different types of G alphas which are present. And depending on their type, they will activate different pathways. Remember that each one of them is generally found in GDP bound form, but when they come in contact with the receptor, they are activated and go into GTP bound form. For simple sake, we can say G1, 2, 3, 4. These 2 are involved in cyclic nucleotide related function. This is with the lipid dependent function, and this is with the cytoskeleton dependent function. One very important thing about the GPCR signaling, that people have been working for this particular protein and the signaling pathway, and multiple Nobel Prizes have been won in the area of GPCR signaling or proteins which are related to GPCR signaling. One of the earliest Nobel Prize was given for the discovery of this molecule, which is cyclic AMP. Then people have been given Nobel Prize for discovery of GPCR. Very recently in year 2012, Robert Lefkowitz and Brian Kobilka received Nobel Prize for their extensive study on beta adrenergic receptor. Which is a GPCR kind of molecule. So basically the 4 types are there, and they have been worked extensively. We also know that people have got Nobel Prize for discovery of small GTPases, Martin Rodbell. So multiple Nobel Prize winning discoveries have been made with respect to G protein, GPCRs and so on. So when we talk about the G protein, we know that in case of GPCR, it is heterotrimeric G protein. It has G alpha, beta gamma subunits, and as shown in this image, G alpha independently tethers to the membrane and G gamma independently tethers to the membrane. When there will be a separation, G alpha will go its own way, beta gamma together will go in their own way. More importantly, the catalytic activity, because in the very first slide I was discussing that GPCRs do not have integral enzymatic activity, rather they associate with proteins which have enzymatic activity. So the protein which they are associating is with the G protein, and out of the 3 subunits, G alpha subunit has the GTP binding and hydrolyzing abilities. Basically binding and hydrolyzing ability towards GTP, what makes it a GTPase. If you think about these polypeptides, they are together and they are active. If you think about these 3 polypeptides, alpha, beta and gamma, when they are together, they are inactive. Then you have the ligand binding region which is there in the receptor. Once ligand binds to the receptor, it brings the conformational change which we have seen in the previous couple of slides back, where we saw that how helices were giving orientation which was allowing more and more G alpha to be tethered and get activated. And once this is activated, beta gamma subunit will be separating and going their own way. Now if we think about the GPCR signaling, I told you the very simple thing, and if you take example of GPCR and if I draw the GPCR this way and the other GPCR this way. So this is inactive, unbound, the ligand is not present. When you have ligand, it goes and binds over here. Now it is ensuring that G alpha GDP is getting converted into G alpha GDP. It is ensuring that G alpha GTP, it is ensuring that G alpha GDP bound form associates with the inactive unbound receptor and gets activated. And I have already told you that this kind of conversion requires GEF, and I emphasize that the intracellular loop regions, or in some sense receptor itself, has the GEF activity. So in the beginning I said that GPCR may present a class of receptor which does not have any integral enzyme activity, but if you consider this GEF activity, then they do have a particular enzymatic activity associated with them. Now this process is done by the receptor itself. This is the GEF activity, and in the reverse direction, if you recollect, for small GTPases or other GTPases, this activity is mediated by gap-like molecule. So in GPCR terminologies, these are normally referred by different names. One of them is called RGS. Basically that is regulator of G protein signaling or G protein stimulation, because it is regulating the stimulated state of the G alpha or GPCR by bringing it back into the GDP bound state. Phosphate is released in this process. There are very unique terminologies which are associated with GPCR, like Often for GPCR, ligand is called agonist. Which will be binding and activating the system. So normally when this G alpha T is activated, you will generate G alpha separate and G beta gamma. Quite often you will see the lot of signaling is dominated by G alpha T, but in some cases we do have examples where G beta gamma also participates in the activity. And what this G alpha T is going to do, depending on which class it belongs to, it will activate next set of signaling and next set of molecules. So in simpler words, this is the situation when ligand is absent. When ligand binds, what you have is the separation of G alpha beta gamma. Don't think that the separation happens independently. This entire complex moves in the vicinity of the receptor. There you will see that GTP has bound. Over here, this GTP bound state actually induced the conformational change which allows beta gamma subunits to go away. Right? Now this G alpha will be signaling and generating the second messenger kind of thing, and generate secondary messenger. So the kind of secondary messenger which is generated downstream of G alpha T suggests that what kind of G alpha we are looking at. So there are three major types of second messengers which are seen as a signaling intermediates in case of GPCRs. First of them is a cyclic nucleotide, whether it is cyclic AMP generation or cyclic GMP generation. If you recollect, when we were discussing the receptor histidine kinase in microbial world, we were seeing something which is called dicyclic GMP. It was a very unique molecule which was serving as a chemoattractant and allowing a sessile lifestyle. Here we will see how generation of cyclic AMP is mediated by a class of G alphas. So there could be two different classes, one which will help in the generation of cyclic AMP or cyclic nucleotide and from ATP, and the other one will be bringing it back to linear AMP. Again, if you recollect from our cyclic di-GMP molecule which was there in bacteria, it was being linearized by two different set of enzymes. So the interplay between cyclic level and linear level of cyclic AMP or AMP molecule is going to be determined by which G alpha is coming into action. The other G alpha, it works through the enzyme which will break down the PIP2. You recollect this, we studied this in case of receptor tyrosine kinase, where the enzymes of PLC gamma family over there, they were breaking down PIP2 into IP3 and DAG. In case of GPCR, this will be PLC beta family which is working and breaking down. And further through this, you will be generating a lot of cytosolic calcium pool which serves as a second messenger. So these signaling intermediates play a very important role, and they are going to divert the signaling in a particular desired direction. So if we look at, there are people have in the researchers have identified different ways to visualize the generation of these second messenger, like in case of certain neurons in the terminal here, cyclic AMP generation. When will cyclic AMP generate? If you give the ligand which is binding to the GPCR kind of receptor and activating the G alpha. So any G alpha which is activating the production of cyclic AMP is classified as G alpha S. S stands for the stimulation of cyclic AMP production. So people have generated molecules which will detect cyclic AMP and fluoresce also. So once stimulus is given, suppose in this case a neurotransmitter like serotonin, once it binds to the receptor, it is activated and you will see lot of signal in this area which can be detected and analyzed further. So there are multiple examples of these kind of receptors which are present and what they are working for and what are the target tissue, like multiple hormones which are working through this cyclic AMP, TSH which is coming from pituitary but works on the thyroid, then ACTH, adrenocorticotropic hormone, works on adrenal cortex, adrenaline works on muscles as well as heart. Depending on the cell type or the muscle type, it will be activating or inhibiting the contraction. Parathormone, which is important for bone activity, glucagon, which is important for the breakdown of glucose, then vasopressin, which is important for kidney function, water retention and so on. Then collectively TSH, glucagon, adrenaline and adrenocorticotropic hormone, they work on fat tissues and mediate the metabolism process. So in this class what we have understood is that GPCRs are the largest family of receptors in humans. There are more than 800 receptors which are known and there are many more molecules, many more genes which code for ligands as well as G proteins. In addition to that, other signaling regulating components which we will see in further classes. And also these receptors are very unique in terms that they associate with heterotrimeric G protein and divert signaling depending on which G protein type has been utilized. The ligand which is called agonist here, it binds to the GPCR, activates it so that G alpha can be activated further, and as an activity of G alpha you generate the second messenger like cyclic AMP. This amplifies the signaling process. Thank you.